Okay, hello everybody. And we're going to carry on our discussion about bacterial growth. Again, this is of interest not only biologically, so that we can understand bacteria themselves better, but because this is also very important clinically, of course. The development of pathogenic populations of bacteria, they follow the same rules that we're going to discuss today. And so it behooves us to understand what those rules are. So the first thing that I want to explain to you is something I have told you before, is that bacteria love surfaces. And many bacteria will, in fact, not grow particularly well unless they are attached to a surface. And one of the reasons for this is that many bacteria love growing in community. And they form what we refer to as biofilms, layers of bacteria bound together, usually by slime layers, in which they can cooperate and in which they actually grow particularly well. So these represent actual microbial communities, not just necessarily of one kind of bacterium either. There may be several kinds of bacteria in these communities which cooperate with one another. Some may be able to break down certain substances that the others can't, and yet they can all pick up the products, etc., etc. So there may be cooperation between bacteria in forming these communities. And so what happens is that they actually form a distinct layer. And this distinguishing feature is usually what binds the bacteria together, which is a slime layer of some sort. But to call it slime is a little bit disingenuous, because the slime itself is actually a very complex compound. It's usually a hydrogel of some sort. That is a substance which forms a jelly in contact with water. Now, the amazing thing about these biofilms is that they're very often set up because, in fact, bacteria can talk to one another. They can actually send messages to one another which are perceived and which elicit a response from other bacteria. And this is particularly important in a process called quorum sensing. And that is that bacteria will often not grow particularly well until there are signals from actively growing bacteria. It's kind of, if you can think of it, it's not entirely true, but think of it in a way as if bacteria are informing one another that conditions are good for growth. And this is accomplished because bacteria produce signaling molecules. They're called inducer molecules, which communicate to other bacterial cells, first of all, attracting them, but also very often bacterial cells will not start dividing, as we'll hear in a minute, until these signaling molecules are present. So these biofilms, here, the schematic diagram of one, you can see all the bacteria here embedded in a slime layer, which is adherent to a surface. So these slime layers don't usually form an impenetrable barrier. They are usually broken up so that water and medium can pass in between them quite easily, providing, of course, nutrients and oxygen or whatever else. And these allow these clumps of bacteria to adhere to the surface, close proximity to one another. They can communicate with one another and share resources. But this also provides some sorts of protection. First of all, it sticks them to a surface and prevents them from being diluted in the surrounding medium. The second thing is it does provide protection, particularly from harsh conditions, but also in living systems, it provides some protection from immune responses, for example. And we find actually that these biofilms are really difficult to deal with in some circumstances, like in clinical settings, because antibiotics do not necessarily penetrate beyond the surface of these clumps if we want to get rid of the biofilm. Disinfectants and antibiotics and things don't actually penetrate that easily. This hydrogel, the slime around them, is actually very protective. Not only that, but these clumps are relatively fragile. They can break up, and a clump of bacteria already active, already dividing, can be released to go and settle somewhere else and found a new branch of the biofilm. So these biofilms are important under natural circumstances. We often find the richest bacterial populations adhered to surfaces. In the natural aquatic environment, for example, you find them all over algae, you find them on the rocks, you find any available surface is usually densely colonized by a layer of bacteria in a biofilm. But there are other instances where biofilms are very significant, and that is, for example, let's just think of ourselves. The inside of our mouth, for example, has a biofilm of our natural microbiota. Our teeth have a biofilm. That biofilm on the teeth, by the way, can be important in tooth decay, because if it cultivates acid-producing bacteria, that is one of the causes of tooth decay. Our nasal passages, the upper respiratory tract, has a biofilm conforming pretty much to this kind of structure. The most important, probably, biofilm of all is that which is present in our gut. From the stomach, from the end of the stomach on down through the entire gut, there is a very, very rich microflora. And there's a huge, huge number of bacteria, and many of them exist in these, will be present, adhering to the intestines, etc., by means of biofilms like this. Okay, so this is not the only place that they are important. In a clinical setting, bacteria can settle on surfaces such as the surfaces of catheters, artificial heart valves, contact lenses. All of these situations may develop very dangerous biofilms. And in fact, 70% of nosocomial infections, a nosocomial infection is an infection which is specifically associated with the hospital or healthcare setting. It's an infection acquired in a hospital or healthcare setting. And 70% of those are associated with biofilms present on medical equipment. So this is very, very significant. And remember, these biofilms are very often resistant to microbicides, to disinfectants or antibiotics or whatever, because they are quite thick. Only the surface exposed bacteria may be killed off. The bacteria interior to the biofilm may be relatively resistant. In addition, the biofilm may set up special conditions. So, for example, anaerobic bacteria, even obligate anaerobes, go back to what we talked about before. Even obligate anaerobes may be able to function in the interior of a biofilm because any oxygen coming into the biofilm is scavenged by bacteria nearer to the surface. So here's why. This is actually a genus that you've probably seen in lab. This is Serratia. Serratia is a common member of our gut flora. And as such, it can easily be an infectant in things like catheters, also those sorts of things. And here it is adherent to a plastic surface of some sort. And you can see here's this thick layer of the cells, and there are all of these strands, many of which have probably been lost in preparation. But all of these strands binding them together, these are all parts of capsules and outer structures outside of the cell wall, forming slime layer, which binds them all together. Okay, so just quick mention that we need always in microbiology to be very aware of what we are culturing and what our relative risks are. And the type of laboratory where we work takes cognizance of all of that. And depending on what you're working on, depending on what you're doing, you will be required to be in a certified laboratory setting with certain levels of safety precaution. So this is the biosafety level. BSL stands for biosafety level. BSL1, this would be your typical biology lab, for example, where you're not dealing with anything pathogenic. Anything you deal with is going to be fixed or harmless. And there's no need for serious precautions beyond common sense. BSL2, this is your microbiology lab. This is your teaching microbiology lab. There may be occasional bacteria which could be pathogenic, but their pathogenicity is very low. And using normal aseptic techniques is sufficient to protect personnel from infection. And there's just ordinary protective gear is used, the lab coat, as you all wear in lab, gloves, frequent hand washing, if need be, eye protection, particularly if you're dealing with transferring broths and things like this, you may wear eye protection. But the risk of pathogenicity is very, very low. So ordinary kind of common sense and aseptic technique, that is sufficient to keep it safe. BSL3, this is a kind of lab where you deal with bacteria or with bacteria or viruses, whatever, which have a recognized risk of pathogenicity, which is fairly high, and where the microorganism is relatively easily transferred and could be acquired or trafficked from the lab to outside. And so these are this would be research work, for example, on many of the clinically significant microorganisms causing illness. And in these labs, the precautions are much higher. There may be restricted entrance to and from those labs. Personnel may be required to wear protective gear depending on the organism. And very often, work is only conducted inside a closed facility, inside a cabinet, for example, or inside special cabinets where the risk of disseminating the pathogen, for example, is very much reduced. And this is BSL3. This would be working especially on things like tuberculosis, for example, or things like that. BSL4 is reserved for highly dangerous and infectious and easily disseminated microorganisms. And this would be, for example, the labs which have been working on SARS-CoV-2, but also on things like Ebola and various other things which are easily transmitted and especially which could easily be trafficked outside of the laboratory. In particular, airborne pathogens of one sort or another. And BSL4 is the level. Very often these labs will require showering in and showering out, donning clothing which is only worn inside the lab and then is discarded or taken for autoclaving and exchanged for street clothes before you leave. The lab is entirely sealed. It is under negative pressure, which means it draws in air the whole time, and exhaust is through HEPA filters, which filter out pathogens. And these are labs which are very, very, very carefully managed under very secure conditions. Okay, So I know that you have encountered culture media under many guises now in your labs. You've seen many different kinds of culture media. Just a few reminders. When we say culture media, what we mean are nutrient solutions prepared so that they encourage microbial growth. In general, we use two kinds: broths that are liquid, it's a liquid solution of the nutrient, and then solid media. And these are liquid media, the broths, which have been solidified. And the commonest solidifying agent that we use in labs is agar. There are one or two special media which use various other kinds of solidification. But in general, we use agar. And if we say that these are sterile media, what we mean is there are no living microbes in them. But once we introduce an inoculum that is a source of microorganisms, we introduce that into the medium, they are no longer sterile. Instead, what we're looking at is a culture. And that is a culture is microbes growing in or on a culture medium. So there are two broad categories of culture media. The first are chemically defined media. Those are media where we know exactly what every component is. We know exactly how much salt there is. We know exactly how much glucose there is. We know exactly how much of every single amino acid there is present, or whatever. So these culture media are often grown either for fastidious organisms. Those are organisms which require special culturing, special nutrients, or else where we are monitoring the metabolic processes, for example, which lead to production of amino acids and things like that. This brings up a point that many bacteria in particular can be grown in an entirely chemical medium which lacks any organic material at all. So they can be presented with inorganic nutrients, and from those inorganic nutrients, many bacteria are able to metabolize all of the biomolecules which they need. As we'll see this in an instant now. But these chemically defined media are often extremely difficult to make. They really have to be very carefully manufactured. So in the ordinary lab, it is usually sufficient for us to use what we refer to as complex media. And these are media where you obviously know what you put in, you weigh everything you put in, but they consist of extracts and digests of proteins and yeasts and things like this, where we don't know exactly what the composition is. We don't know exactly how much tryptophan there is in the medium, for example. But we know that there's plenty enough tryptophan for everybody to grow. We know there's plenty of amino acids available. We know there's plenty of vitamins, essential vitamins for everything to grow. And these are the kinds of media we usually label as nutrients. So a nutrient broth, a nutrient agar, are actually complex media. And a nutrient broth, for example, usually consists of a yeast extract that is cultured yeast, which has been killed, and then it is maybe partly digested by enzymes. But the yeast extracts are useful because they not only provide a source of amino acids, but very importantly, they provide a source of vitamins. Then we provide a rich amino acid source, and this is usually something you'll see that if you look on your plates and broths, you'll see that often they will say that they are peptone, they have peptone in them. All that peptone is meat, which has been digested by pancreatic enzymes. And the same for tryptone. These are just enzymatic digests of a rich protein source. So the protein gets chopped up into individual amino acids or into small dipeptides and things like that that are easily assimilated by bacteria. So, and then a nutrient agar is simply this plus agar. And we'll talk about agar in a sec. This agar is extracted from seaweed, and it's a very complex polysaccharide. We use it in most media because there are very few bacteria, unless they are from marine systems. There are very few bacteria which are able to break down agar. So it is maintained as a solidifying agent. If we use with gelatin, for example, we can also solidify with gelatin, but gelatin is an animal product. It's jello, right? And it's an animal product, and many bacteria digest it very, very easily. And so we tend not to use that, because you place onto a gelatin plate and you come back the next morning and the whole thing is liquid because they've broken down the gelatin. Place onto agar, very few bacteria can produce an agarase, which will break down the agar. So the great advantage of agar is that it liquefies at boiling point, around 100 degrees Celsius. But it only solidifies at around about 40 degrees Celsius. This is introduced a very useful property to agar. And that is this temperature here, 40 degrees, most bacteria can survive at least briefly at this temperature, 40 degrees Celsius. If we should want to mix bacteria with the agar when we pour a plate, for example, as we'll see in a minute, we can use this as a counting technique. We can do a pour plate in which we take a unit of agar, of nutrient agar. We introduce a known quantity, a known volume of bacteria, of medium with bacteria in it, for example. Pour that as a plate and count how many colonies grow. That enables us to determine a bacterial population. So this is just another useful property of agar. Okay, so here is a typical complex agar. This is what you're using in it. This is the sort of thing you're using in lab the whole time. So here you are. This is peptone. This is partially digested protein there. Beef extract. Beef extract also provides vitamins, but it also provides various minerals and trace elements and things like that. Sodium chloride. We don't always include sodium chloride, but it is included in some recipes. And it's usually just to provide a balanced osmotic environment. Agar, the typical use of agar is 15 grams per liter. That is for a solid agar. We sometimes make a sloppy agar for various purposes, which is less, but this is your standard for the plates that you're using in the labs, 15 grams per liter and a liter of water. That's a simple, easy recipe for a very good complex nutrient medium. But here's a chemically defined medium. And this you can grow many bacteria on. They don't grow as fast as they do on complex media. But for example, even Escherichia coli, which is a normal, typical chemoheterotrophic bacterium, grows extremely well on complex media. But here we can grow it on this kind of medium as well, where we know absolutely to the milligram how much glucose there is present, for example. We provide a rich nitrogen source, but not an amino acid source. Here it is ammonium phosphate here. Then various other compounds which provide all of the molecules which a living organism needs in its biomolecules. But notice there is nothing organic here. There's not a single organic molecule. And yet on this kind of medium, Escherichia coli can manufacture everything that it needs, provided it's got the basic atoms provided to it in an available form. It can produce everything that it needs. It can produce all of its amino acids, all of its vitamins, everything. There are other organisms, other bacteria, other organisms which might require, for example, the addition of some vitamins. And those are called the essential vitamins. And a vitamin is essential if an organism cannot produce it on its own. But many bacteria are quite happy growing in a chemically defined medium like this. Okay, there's some special instances that we do need to talk about as well. And look back and see that our bacteria have different relationships to oxygen, for example. And if we want to grow any of those bacteria, which have special relationships to oxygen, for example, they are obligate anaerobes. And then we may need to produce a medium or conditions in which oxygen is not present in the medium at all. And there is a way to do this even for media that we just use on the lab bench. And one of the ways is to introduce into the medium a substance which will bind all the available oxygen. So there is none of free in the medium. And the most common way to do this is to use this substance here, sodium thioglycolate. And it binds all the oxygen present, so none is available free in the medium. We heat, usually with that, we heat it up, first of all, to drive off any small amount of oxygen that might be present. Then we allow it to cool, and we usually take precautions not to disturb it, not to shake it around or anything. And when we do that, you see that often there is a little area at the very top. Let me just go back here. There's often a little area right at the very top that might have a little bit of oxygen in. The rest of a tube, for example, would be completely anaerobic. To check what oxygen is, whether oxygen is present or not, we can introduce into that medium as well an indicator, which usually it's a substance called resizurin, and it turns pink in the presence of oxygen. In a typical thioglycolate tube, you'll see a little thin band of pink right at the very top of the tube, and then the rest of the tube is clear, indicating that it's completely anaerobic. Here's another way to grow anaerobic bacteria very easily, and that is we use an anaerobic chamber, and you'll see the use of one of these. These are just a small chamber. We put our plates and whatever inside the chamber, and then we introduce a packet of chemicals, which actually, there are various ways that they can work, but the commonest is that they bind all of the oxygen to hydrogen, and they they create water, but they use up all of the available oxygen. Again, we usually include an indicator. Methylene blue is the commonest one. Methylene blue is only blue in the presence of oxygen. When there's no oxygen present, it turns colorless. So this is a little anaerobic chamber, but you can actually use a very big anaerobic chamber. This is an anaerobic chamber, which is typically used for fastidious obligate anaerobes, such as Clostridium. The Clostridia are very sensitive to the presence of even very small amounts, very small amounts of oxygen. And these anaerobe chambers, the interior is completely anaerobic. There's no oxygen present at all. You use gloves and sleeves so that the interior here is never open to the outside at all. And there is an airlock here. So if I want to introduce anything into the interior here, I have to store it in this airlock, which is evacuated and replaced with nitrogen. That is repeated several times so that we wash out any oxygen that is present in the airlock and in the medium and everything that we've introduced before we move it in to the interior of the chamber. Here's the last one to mention. Now, this is, we didn't talk about this when we talked about anaerobes and aerobes and everything else, because this is a group of organisms which require high carbon dioxide levels. Now, this applies quite often to pathogens. There are certain of the pathogens which are well known for being capnophiles. They like to have high CO2 conditions in order to grow optimally. And the one that I would mention to you is Neisseria. Neisseria gonorrhea is the causative agent of gonorrhea. Neisseria meningitidis is the cause of meningitis. And when we grow them, they grow best in capnophilic conditions. Well, you can also use a little chamber like this and get a special envelope that sets up capnophilic conditions. But there's a very easy way to do it, and that is the old-fashioned way. It's still done because it's so simple, and that is to burn a candle. It's called a candle jar. And so we introduced all our media. Here they are. We stand a little candle, a nightlight or a yahrzeit candle on top, light it and close. It will use up a lot of the oxygen, not all of it though, but it will produce abundant carbon dioxide, and that's ideal for capnophiles. All right, so just you will already have encountered some of these other media in lab already, but it's good to explain them exactly so that you understand all of the theory behind it. We don't only use media for growing bacteria. We also use media as a diagnostic tool to demonstrate different biochemical properties of the bacteria that we are growing. The first kind of media that we would suggest to do this are selective media. And selective media suppress the growth of bacteria that we're not interested in, and they encourage the growth of bacteria that we want to grow. And to do so, they usually contain inhibitors which suppress growth. Sometimes a selective medium may lack a particular nutrient, which our bacteria of interest don't need, but everything else does. But that's a special circumstance. Most commonly, we include substances which actually suppress growth of things that we don't want. So that's selective media. A differential medium is one which allows different things to grow, but they grow with different properties. They display different properties on growth so that we can distinguish between them. And these are differential. And many of media that we use are both selective and differential. They can be either, but many of them are both, as we'll see in a minute. So here is a purely differential medium. This is sheep's blood agar. Now, sheep's blood agar is a very good general nutrient medium. And really, it will encourage growth of most bacteria that we place onto it. But it is differential. And it is differential because different bacteria produce different reactions in the blood that is present in the medium. This blood medium is made, specifically made, so that the blood corpuscles in the medium remain intact together with their hemoglobin. And different bacteria do different things to those blood corpuscles. Here's the bacterial colonies there, the little dark dot there. This clear zone shown around here is a particular reaction to the presence of those bacteria. The bacteria are releasing exoenzymes, as all bacteria do. They're releasing digestive enzymes which percolate into the, diffuse rather, into the medium. And they're doing something particular to these blood corpuscles. In this case, they're producing this clear halo around. Not all bacteria will do that. So this here is called hemolysis. Hemolysis literally means the breaking of blood. And that is what is happening here. The blood corpuscles are being broken down, but they're being broken down in a particular way. There are three classic patterns of hemolysis. The one that we're seeing here, the absolute clearing, is called beta hemolysis. And here it is here. Here's a bacterial growth that's been traced out in the shape of the beta symbol. And you can see it produces an absolute clearing. And that is because it not only lyses the bacteria, the blood corpuscles, it also completely breaks down the hemoglobin and uses it. It absorbs the component parts. It produces absolute clearing. The second kind of hemolysis is called alpha hemolysis, and it's a little bit more difficult to discern. Here it is here. Here's an alpha symbol grown, and if you look carefully, you can see the hemoglobin has turned green. The blood corpuscles are not lysed, and the hemoglobin is only partially broken down, and it instead forms of being red. It forms this green pigment, and the green pigment is associated immediately, usually, with the growth. It's often easiest to see if underneath of the plate, below the growth. It is a distinct green look, tells you that's alpha hemolysis. The third one is gamma, and here it is here. This is gamma hemolysis, and gamma is nothing. There is no reaction. There is no reaction of the blood to the presence of bacteria. So these you need to know. Alpha, beta, and gamma hemolysis, three distinct reactions. And these are diagnostic for many bacteria. These are particularly important in cocci and especially in streptococci, but also in staphylococci as well. But in streptococci, they're a classic diagnostic tool for pathogenic streptococcus. So here's another differential medium. This is one you will encounter in lab. And this is mannitol salts agar. And mannitol salts agar tells you exactly what's in it. Mannitol is a sugar, which some bacteria can ferment and others cannot. The second component is salt. Mannitol salts agar has a high proportion of salt in the medium. And that is very strongly selective because there are not many bacteria which can withstand the osmotic pressure induced by high concentrations of salt. But there's one group which can, in particular, and these are the staphylococci. And just think about it. If I was to ask you to derive a culture of staphylococci, where would you go and look? How would you do it? I could do this in lab. I could give you each a nutrient agar plate and a swab and tell you, okay, now go find me some staphylococci. And the easiest thing to do is to swab your skin because the skin is very rich in staphylococci. The staphylococci are part of our normal microflora. It's one of the reasons why it's a very bad idea to go and douse yourself with antibacterial soaps. We have a natural flora on our skin, which is healthy. And part of that flora are the staphylococci. There are some of those staphylococci which are potentially more pathogenic than others. And one of them is Staphylococcus aureus, that is shown growing here. But there are others which are completely benign and seldom clinically relevant. For example, Staphylococcus epidermidis, and that is shown growing here. Here are the typical reactions for mannitol salts agar. First of all, Staphylococcus grows happily on mannitol salts agar because it's resistant to salt. If you want to know why, lick your skin and taste the salt. The salt is introduced from your sweat. So that Staphylococci and other microflora are resistant to the salt in your sweat. Other things are not. Here it says here, this sector here of the plate, it says uninoculated. I could as well have inoculated this with E. coli and it would not grow. There are many, many other bacteria I could inoculate here that would also not grow. Mannitol salts is strongly selective for things like the Staphylococci and selects against things like E. coli, those sorts of things which don't live in a high salt environment. So it's selective, but it is also differential because it contains mannitol, and not everything that grows on mannitol salts agar can ferment mannitol. The medium also contains an indicator, and the indicator turns yellow if, just give me 1 second, please. Sorry, there was a cat fight. Okay, I've forgotten now. I've completely forgotten where I was. Okay, I told you. Selects for the things like the Staphylococci, but distinguishes between the Staphylococci because not all of them can ferment mannitol. Staph aureus does. It produces this typical yellow color on mannitol salts agar. Staph epidermidis cannot ferment mannitol, and it does not produce that color. So this is both selective and differential. Here is another, this one you have also encountered in lab, and this is a very important clinical medium called McConkey's agar. And it, again, is selective and differential. McConkey's agar contains crystal violet, a dye that you've already encountered, and it also contains bile salt. Now, the importance of bile salt is that our gut is flooded with bile. So all of the gut-associated bacteria, all of the enteric bacteria are resistant to the presence of bile. The vast majority of those bacteria, of enteric bacteria, are gram-negative. And bile salt is strongly selective for gram-negatives and against gram-positives. So the same thing, by the way, is for complex reasons. The same thing is true of crystal violet. It selects against gram-positives. So this is a medium which is selective for gram-negatives, and it is also differential because it contains the sugar lactose. And it contains also an indicator. So if anything ferments lactose, it will have a yellow color around it. So McConkey's is selective and differential. That's why it's so important in the clinical lab, because it is one of our ways that we can distinguish lactose fermenting enteric bacteria, usually associated with fecal contamination or with feces. So here's McConkey's agar, and here the first experiment they've done here. Here's Escherichia coli, and E. coli will ferment lactose, and it turns this deep purple color here because it ferments lactose. If we grow Staphylococcus, try to grow Staphylococcus fails to grow. It is gram-positive, and it is inhibited by the bile salts. Here are two different enteric bacteria. Here again is E. coli, deep purple because it ferments, but here is Salmonella. If it ferments at all, it ferments very, 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 very weakly. And you can see it's mostly just this colorless color here. The yellow behind is not clinically significant. The point about this is it's not fermenting lactose the way that E. coli is. We are differentiating between E. coli and salmonella. If you were to look at salmonella and E. coli under the microscope, they look pretty much identical. They're both gram-negative rods, and they grow pretty much the same in very difficult to tell just looking at them what the difference is. But here is a clear difference. E. coli ferments lactose. Salmonella does not ferment lactose. Okay, so I'm not going to run through. This is a summary slide for you of the different kinds of media. Make sure you understand all of those. You will surely be asked at some point questions about this. Okay, now let's just have a look at the way in which these bacteria populations actually grow, because there are distinct patterns of growth which we need to understand. We know a characteristic of the bacteria is that they grow by binary fission. That is, they grow to a certain size, and then the cell divides equally to produce two daughter cells, which are smaller. And those two daughter cells then carry on growing until they reach a requisite size, and then they also divide. So what we're looking at under ideal conditions, we're looking at repetitious duplication of the population. We have a population of bacteria at a certain, they will grow, and at a certain point, they all divide, and we double the number. So every time cell divides, you double the number. This gives a very important property when we look at whole bacterial populations. Sorry, just a little discussion about how they divide, but I think we've already done this when we talked about cell function. Okay, so that from cell to growth to division into two new cells, that is called the generation time. The generation rather, and the time it takes is the generation time. That can be extremely short. In E. coli, for example, if we're growing under ideal conditions, that's in abundant nutrient, at 37 degrees Celsius, which is the optimum temperature for E. coli, it's as little as 20 minutes from an individual cell to that cell reaching a size big enough for it to divide and produce two cells. Now, when we follow those two cells, we're going to repeat the 20 minutes. Okay, they're also going to take 20 minutes, and then we're going to end up with 4 cells. So we're going to double our population size every 20 minutes. The total number of cells that we're looking at, apart from the very first cell that we start with, if we want to work out the total number of cells we take, it is 2 to the power of the number of generations. Okay, so if we take our 1 cell and we double it in about 20 minutes, we end up with 2 cells, right? That's 1 generation. We take now 2 generations. Those 2 cells divide, 2 to the 2, 4. That's how it works. When we take that ideal part of the growth, doubling every generation time, that produces a mathematically distinct pattern of growth. And that is that the growth is logarithmic. If we plot it out arithmetically, we end up with a curve. And if we plot it out logarithmically, we end up with a straight line. I'll show you in a minute. So that is a particular phase of growth. We're watching as these cells begin dividing. First of all, we start off 1, then we get 2, 4, 8, 16, 30, etc., etc. And when we represent this visually, this is what we end up with. We end up with this curve. This is an exponential curve. But we refer to it as log growth, logarithmic growth, because if we plot the log of each of these numbers, the log to the base 10 of each of these numbers, as we're doing here. So we're plotting 32, 1,024, 32,768. We end up with these log numbers, 1.513, 4.5, etc. We plot those, we end up with a straight line. A straight line is the representation, logarithmic representation of exponential growth. Here are the number of generations on the bottom. Here is the actual number of bacteria here on this growth here. But if we look at the log numbers, we end up with this straight line here. That is very, very typical for bacterial growth under ideal conditions. But it does not represent the entire growth pattern that a population of bacteria will present. We're talking about a very special situation, however. We're talking about a bacterial population which is growing in a finite medium. That is like in a broth tube, right? Or in a particular area of the body or something like that. We're talking about bacterial population which is being given a set amount of nutrients, etc. And we're going to watch what happens to that population. So what we're going to do is we're going to take our typical nutrient agar tube, for example, and we're going to introduce a very small number of bacteria into that nutrient agar tube. And we're going to follow what happens. Well, here, this, by the way, is log there. That's the log scale. This is time in hours. So as we would expect, there's one point here, there, where the growth is logarithmic. If we were to do this arithmetically, what we would see is an exponential curve, but we are plotting logarithmically. So here it is here. That we've already seen. That is this. That is this phase of growth. But there's a phase which precedes it. And we've introduced our bacteria. We didn't introduce necessarily a lot of bacteria into our tube, but there are certainly bacteria there. And yet we see virtually no bacterial growth for a long time. And this is called the lag phase. What is happening here is not only are bacteria adjusting to conditions that surround them, but they are emitting inducer molecules. They are talking to one another. This is an example of quorum sensing. The bacteria are not going to begin growing until the signaling molecules are sufficient that it sparks growth through quorum sensing. Once they begin dividing rapidly. As time passes here already, they are passing into exponential growth. When the whole population is dividing, let's say it's E. coli dividing every 20 minutes, the whole population is doubling every 20 minutes, then we are in exponential growth. But remember what I said, this is in finite conditions. This is in our growth tube or whatever. Nutrients is being used up. Excuse me. Nutrients being used up. Maybe oxygen is being used up as well, and carbon dioxide is building up. Waste products are being released by the cells, and conditions begin to deteriorate. They pass away from ideal. When that happens, the cells begin to slow down. Growth rate slows. And in fact, cells begin to die. We enter a period of equilibrium where as many cells are produced, die. The population level remains constant. And this is called stationary phase. Even that can't continue forever because now the conditions become positively hostile. very little nutrient, high levels of waste, etc., etc. And now more cells die than are produced, and fewer and fewer and fewer are produced, and more die. In fact, they do so at a logarithmic rate again. And this is called the death phase. Now, it seldom passes down to absolute zero in actual fact, but it can do if you keep it long enough. But there may be a few cells here that are able to remain alive, often by reducing their metabolism tremendously. We often observe that they lose biomass, they become very, very small. But if we take those cells and put them into rich medium, they can revive. And these are called persister cells. And in clinical situations, persister cells are very important because they can, for example, reestablish infections if this was a pathogen that was growing. Okay, so this is the typical pattern, the bacterial growth curve: lag phase, log phase, stationary phase, and death phase. So you need to understand this really carefully. All right, so now I'm very quickly just going to talk to you about different ways that we have of actually assessing bacterial populations. This is often very important. You've already seen an instance where we started to do this for that water sample. We will look in that water sample from the East River that you're doing in your labs. You're looking at a specific population. You are looking at a population of looking for a population of gram-negative lactose fermenters. Why are we particularly interested in those? Because they are indicators of fecal contamination. We take them as indicators of fecal contamination. Okay, gram-negative lactose fermenters, that should be familiar to you from what I told you about McConkey's agar, for example. So we often want to know how many bacteria are present under certain circumstances. We also may want to know how many of a particular kind of bacterium is present. And so there are various ways, some of them fairly complicated. I'm going to kind of skip through fairly quickly. If you want to know more about these, go to your text and read up about them. Okay, so there are ways to directly count bacteria. And classically, these are the ones that we use: either plate count, filtration, most probable number, which should ring a bell, or by direct microscopic count. So to do a plate count, what we do is we will take a sample, let's say our water sample, and we have no idea how many bacteria are present in that water sample. But if we were to take a part of that water sample, let's say 0.1 mil, and plate it onto a plate, spread that little water sample out all over the plate so that bacteria were well separated from one another. Each of bacterium separated from the others would grow to form a colony. We could count those colonies, and then, because we know of a dilution, we took 1/10, right? We took 100 microliters. That is 1/10 of a milliliter. And multiply the number of colonies, C, by 10, and that gives us the count per milliliter. So that's fine, as long as you've got few enough bacteria that when you put your sample on the plate, you have between 3 and 300 colonies to count. Between 30 and 300 colonies to count. That is the statistically viable count. And those are called colony forming units. You have 30 to 300 colony forming units on your plate. That is statistically viable. Less than 30, it's too few. More than 300, you're going to have great difficulty counting accurately, especially because colonies will be growing on top of one another or into one another. So if we've got few enough bacteria in that 0.1 mil, that we get between 30 and 300 colonies growing, well, that's fine. If we have that few bacteria, that we're doing well. But very often we have far more bacteria than that. We may have thousands, millions of bacteria per 0.1 mil. We are going to have to actually dilute our original sample somehow. Then plate that. The key is we always keep a really careful record of what our dilution is. As long as we keep a careful record of the dilution, when we end up with a countable plate, we can work backwards and work out the complete count. So there are two ways of doing this. The first, I've briefly mentioned, you can actually make an agar solution, cool it to around 40 to 45 degrees, put in your sample, your dilution sample, mix it up well, and pour the plate and count how many colonies grow. The other way is to take an actual subsample of the water and put that onto an already set agar plate and spread it over the surface so that it's well spread, as I mentioned before. And that is called the spread plate method. The key to all of these is that we need to do serial dilution in order to get there. So, for example, you will do this in lab, so I'm not going to labor the point. But have a look here. We're going to set up a series of tubes, and we're going to do a serial dilution. We're going to do a dilution series. Here in this instance, it's a very easy one, because what you're going to do is going to take 1 mil of your original and put it into 9 mils of whatever this is, just water, for example. That dilution is 1 mil, 9 mils, that's 10 mils total. So it's 1 mil in 10. That's a 1 in 10 dilution. Properly represented in scientific notation, that is a 10 to the minus 1 dilution. In a serial dilution, what we're going to do is we're now going to take 1 mil of this dilute solution. We're going to take 1 mil of that, and we're going to put it into 9 mils there. That is a 1 in 10 dilution of a 1 in 10 dilution. You multiply them. 10 to the minus 1 times 10 to the minus 1 is 10 to the minus 2. It is a 1 in 100 dilution. In this dilution series, we're now going to take another mil. We're now taking 1 mil of our 1 in 100, and we're putting it into 9 mils. That is going to now be a 1 in 100 dilution of a 1 in 1,000 dilution. What is that? That is 1 in 1,000, 10 to the minus 3, and so on and so on. In this instance here, we are doing a 1 in 10 dilution series, but remember that each time we do so, we are doing a 1 in 10 of an already dilute solution. This is why you need to keep a really careful record of what your dilution series is. As long as you know what your dilution series is, it's quite easy to work out. Now what we're going to do is we're going to plate 1 mil of those. You won't do 1 mil in lab, but just for this instance, we're going to take a mil. We're going to put it onto a plate there. We're going to take a mil of this dilution, put it onto there. We're going to take a mil of our 1 in 1,000, which is 10 to the minus 3, going to plate it there. 1 in 10,000, 1 in 100,000. Remember the rule. We need a plate which is between 30 and 300 colonies. Here it is here. It's our 1 in 1,000 dilution. And you can actually count the number of colonies that are present here. You'll see how they decrease in number. Here, they're far too few to count. This one may be, I'm not quite sure, this one may be countable, as well as this one here. But definitely, this one is between 30 and 300. You count the number of colonies. Let's just say we have 27 colonies. Well, we've got 27 colonies that are produced by a 1 in 1,000 dilution. What you need to do, you take your 27 and you divide it by 10 to the minus 3. You divide it by 1,000. And what do you end up with? 27 colonies, 2.7 times 10 to the 4. Do it. Take your calculator or whatever, but you should be able to do it up here. Divide 27 by 10 to the minus 3 and see what you end up. You will end up at 2.7 times 10 to the 4, 2.7 times 10 to the 4 per milliliter. That is your population size. Okay. If you lost, go back through everything that I've said, or else just look at this diagram and work it out. At each dilution, work out each dilution, and then look at these plates. Just take my figure, say 27 here. Divide 27 by 10 to the minus 3, and that gives you your total population. The population is usually represented as colony forming units, CFU per milliliter. We'll do this again in lab, so don't panic. This is what it looks like using our different methods. This is our pour plate here. Here's our water sample. This is our dilution, right? You can either take take whatever you decided to use in using our preset diagram, 1 milliliter. You can take 1 milliliter, mix it up with agar, spread it out very, very well, and let it set, and then count the number of colonies. That is a typical pour plate. A spread plate, we use already set agar, put our 1 mil on here. Actually, you usually use 0.1 mil. In the previous diagram used a mil just to make the dilutions easier. But here we're using 0.1 mil of the dilution. Spread it out until all the water is absorbed by the agar, and then your bacteria well spread, and then count the number of colonies. That is the spread plate method. There's another way to do this, and that is you can pass a water sample through a filter. This is very commonly done in water quality testing. So you can pass a dilution or a raw water sample through a filter, which will trap bacteria. And you then take that filter and you put it onto agar. You can very often they will use something like McConkey's agar and count the number, for example, of pigmented colonies. That will tell you the number of coliform bacteria, for example, that are present on that filter. There's other ways. The other way that we can talk about, you have already heard about, and that is the most probable number method. Now, most probable number method is backed up by a huge body of mathematical theory, which we are not going to discuss, thank heavens. But it is a standard method of getting an approximate population of bacteria, either total population or else specific kinds if we set up particular culture conditions. So hold on 1 second, something's being closed off here. But we have done this in lab. Most probable number method depends on setting up culture tubes and then seeing with a dilution series, you usually do 10 mils of a water sample, 1 mil of a water sample, and 0.1 mil in 5 different tubes, and seeing how many of those tubes are positive after growth, after you've set aside a time for growth. And then looking at a most probable number table and getting a number. Very importantly, most probable number gives you bounds of confidence on either side of your estimate. That is what you've done, you have seen in lab. But we can also count bacteria by looking at them directly. We can use a microscope to actually count. So this depends on a special microscope slide called Petrov-Hauser cell counter. And this is a microscope slide which actually sets up a little chamber with a very specific volume in it that you are observing through the microscope. So here's how it works. There is an area, this here, the center part here is actually lower than these on either side. You use a special cover slip, which is rigid, doesn't bend at all. So you're going to put it, you take a water sample and you put it here on this lower part. You put the slide over the top, the cover slip rather, over the top, and it creates a little chamber underneath here. It has a certain depth, which you know, and it has a certain area, which you know, because when you look through the microscope, this underneath here has a grid on it here. So what you're looking at is a set volume. If you know how many squares you're counting, you know the area you're counting, and you know the depth. Therefore, you know the whole volume. And you count the number of bacterial cells that you see in that area. In whatever area you choose, you count the number of squares, you know the area, you know the depth, you know the volume. And you end up with a direct count of the number of cells per volume. There are a couple of rules that you have to obey. You make a rule for yourself. I won't count anything which is touching this line here or is touching that line here, there. Instead, if something is over there, then it doesn't, etc., etc. There are a couple of rules just to stop you from double counting. Usually, we look away and move the stage randomly so that we encounter a certain number of different areas to do a count and then do a knee. That is a direct count. You can read through all of the theory here. There is another way which is very frequently used in the lab. We won't do it in lab, but in a clinical lab, it is very, very commonly done. If we want to know what a total population of bacteria is, we can use a spectrophotometer. And what a spectrophotometer does is this is a light measuring device. If, for example, I have a tube of medium, which has no bacteria growing in it, and I put it into the spectrophotometer, the spectrophotometer will measure how much light is absorbed by this tube. If there's no, I know that this is sterile, I can set that as 0. I can set my spectrophotometer to measure that as 0. That we call the blank. Now, if I take a tube, the same medium in which I've been growing bacteria, then if I put that into the spectrophotometer, the bacteria themselves are going to scatter and absorb light as it passes through. Less light is going to pass through this tube than that tube. And I can measure how much light is actually, what that difference is. And from that, I can infer something. The more bacteria that are present, the more light is going to be absorbed. The less light is going to be passing through, right? That stands to reason. But that doesn't tell me how many bacteria are there. In order to understand what this difference means, what this figure that I'm measuring on the spectrophotometer means, I have to actually do a separate set of experiments. What I have to do is I have to have some other way of measuring the actual population. So, for example, I might do it by direct count, looking through a microscope, right? That's one way. I might do it by plate count. So I can say, okay, at this particular spectrophotometer reading, there are actually this many bacteria present. So, for example, I can say here is I can set up an experiment where I measure how many bacteria I have and how much light that they absorb at that population level. This is called the standard curve. Now, this would be a huge nuisance if you had to do this every time you wanted to use the spectrophotometer. But fortunately, you only have to do it once for a particular bacterium under particular conditions. In a particular medium at a particular temperature, you only have to do it once, produce one standard curve. Now, whenever I go and measure the spectrophotometer reading, I see how much light does a particular tube absorb. I can go to my standard curve and I can say, okay, at that absorbance there, this is how many bacteria I have per milliliter. In fact, for well-known bacteria like E. coli, you can look up this standard curve. You can actually get a reference that will give you that standard curve, so that then if you're growing bacteria in the lab, all you need to do is measure the absorbance, and you can refer to the standard curve to get your final population. All right, so that's it for this class. Remember, there is one more class to follow this week. That will be out on Friday. Thank you.